Yes, everything is okay.
Great.
You can start.
Thank you very much. So today I will be talking about some recent research that we are doing in the lab, which has to do with the molecular mechanisms of Alzheimer's disease pathogenesis. So what essentially we are interested in studying in the lab is long-term homeostasis in neurons, in neuronal cells. So we want to understand how these cells, which are non-dividing, long-lived cells, actually they will last for the entirety of our lives. Some of them are even born before we were born and will literally die after we die. They have to maintain their homeostasis for tens of years, even more than 100 years, if we are lucky. So they face enormous challenges when it comes to maintaining their function and also sustaining their energy needs. Because neurons are, as you know, highly energy consuming cells. Even if they are idling, they are consuming a lot of energy just to maintain polarization of their plasma membrane. So they depend almost exclusively for their energy on mitochondria, the energy producing organs of our cells. These cells cannot perform glycolysis to the extent, for example, that muscle cells can do. So almost entirely their energy comes from oxidative respiration, oxidative phosphorylation in mitochondria. And if mitochondria are not capable of providing this energy needed for neurons to sustain their function, then of course we have pathology. And one of the major hallmarks of Alzheimer's disease is actually the accumulation of damaged or dysfunctional mitochondria in the neurons of these patients. And this had been observed several years ago. It is actually a common denominator, one of the common features that we observe in the neurons of Alzheimer's disease patients. So we wanted to understand what is bringing about this phenomenon of accumulation of damaged mitochondria, which interestingly enough is not just the feature of neurons that are degenerating in the context of Alzheimer's disease. It is also a characteristic of cells in the context of many other pathologies. Some of them are actually related to aging, so their prevalence increases with age. For example, we do see similar accumulation of mitochondria in cardiovascular diseases, in ALS, in Parkinson's disease, Huntington's disease, and many other disorders. Some of them have been linked to the aging process itself. And what made it even more interesting for us is that this phenomenon of accumulation of damaged mitochondria that is observed in multiple pathologies is also, as it appears, a common and conserved feature of the aging process itself. So aging is accompanied by accumulation of mitochondria. You can see this in two very distant organisms. On one hand, we have rats, a mammalian experimental model, where we can observe this accumulation by electron microscopy. We can even count and quantify the number of mitochondria in the cells of these animals during aging. And if we compare animals of 6 months to animals of 24 months of age, as you can see, there is almost a doubling in the number of mitochondria in the cells of these animals during aging. We can even observe this phenomenon in vivo in live animals of C. elegans. C. elegans is a small nematode worm that is used as a model organism, as an experimental animal when studying processes like aging. And we can even very conveniently monitor this accumulation using specific GFP reporters that localize in mitochondria. So the intensity of fluorescence in this case corresponds to the number of mitochondria in the tissues of this animal. And if we compare animals of day 1 adulthood to day 10, This is already quite old for these animals that live for about 15 days. So they have a short lifespan, and this is making them convenient for such studies. Still, we see this accumulation of mitochondria. So this is a conserved feature of very distant organisms, suggesting that there must be a conserved fundamental mechanism that governs the homeostasis of mitochondria in very different organisms. And this is happening during aging in both these systems. But it is intriguing that actually in mutants of C. elegans that show extended lifespan, and we have such mutants in the worm where lifespan has been extended, for example, by lowering insulin signaling using specific mutations in genes that encode insulin receptors. DAF2 is one such gene, or TGF-beta signaling. Lowering TGF-beta signaling again extends the lifespan of these animals. So DAF7 is the TGF-beta of the worm. Now, these two strains are long-lived. And contrary to what we see in wild-type animals, where we do see this accumulation of mitochondria during aging, in these long-lived genetic backgrounds, we don't see this accumulation. So there appears to be a link between longevity and the non-accumulation of mitochondria during aging. In this case, we can very easily observe and quantify mitochondria using MitoTracker Red, a dye that stains mitochondria in these animals. So this suggests that maybe there is a causative relationship, because up until this point, this is just a correlation. What we see in wild-type animals is accumulation of mitochondria. In long-lived mutants, these mitochondria don't accumulate. So is this just a decoration of the aging process, or is this causative to aging, this accumulation? Now, to address this question, of course, we would need to be able to adjust, to change mitochondrial content at will, and then observe what happens in terms of lifespan in these animals. And in C. elegans, actually, because it's a genetic system, we can do exactly that. We can manipulate mitochondrial content using genetic mutations in specific mitochondrial proteins. If we do that, we can exacerbate the accumulation of mitochondria in wild-type animals, or we can ameliorate the accumulation of mitochondria in long-lived mutants, where accumulation is already less than in wild-type animals, if any. So what we observe is that whenever we actually make things worse in terms of accumulation of mitochondria, we exacerbate the accumulation of mitochondria, we see that there is a shortening of lifespan. But in contrast, when we lower the accumulation, we make accumulation of mitochondria even less, for example, in long-lived mutants, then we extend the lifespan of worms. So there appears that indeed there is a correlation and a causative relationship between accumulation of mitochondria and aging. Whenever we enhance this accumulation, we shorten lifespan, we make things worse in terms of longevity. And whenever we block this accumulation or make it less pronounced, we extend lifespan, we promote longevity. So, of course, now the question becomes, what controls the number of mitochondria in cells, and how does this mechanism change or fails during aging so that we get this aberrant accumulation of mitochondria? If we consider what might be involved here, what mechanisms might be at play, there are two main mechanisms involved. On one hand, we have mitochondrial biogenesis, the process that creates new mitochondria in cells. And on the other hand, we have mitochondrial degradation, mitochondrial turnover. Now, these two processes, as you can imagine, are in balance with each other in normal functioning young cells. Of course, there is some room for adjustment depending on physiological conditions and energy demands, but always there is a balance restored between these two processes. However, this balance collapses during aging or in the context of pathologies, associated with aging, and we end up with runaway accumulation of mitochondria. Now, this could be either due to increased biogenesis or decreased degradation, or both, of course. And we wanted to understand what exactly is the molecular mechanism that controls these processes and how they are coordinated. So we resorted to genetic screenings to identify the genes that are involved. And in the nematode, we can do these experiments easily, and that's why we chose this organism to dissect this process before going to more complex systems, such as mammals, for example, or even primates. So first, we screened the genome of C. elegans for such genes, either using forward genetics, unbiased approaches, or reverse genetics using RNAi libraries that are available for each and every gene of C. elegans. So by knocking down each and every gene of the worm, we identified those genes that are involved in this process, in regulating the number of mitochondria in cells. And through this genetic screening, I'm not going to describe it in detail in the interest of time, we identified 3 proteins. The first is DCT1 NIX, the second is PINK1, and the third is parking. These are 3 proteins. I'm going to explain what they do. They are all involved in a process called mitophagy. So this suggests that mitophagy is involved in the regulation of the number of mitochondria in cells. But what is mitophagy? Mitophagy is a specific type of autophagy that targets mitochondria. So we have, as you know, multiple types of autophagy. Major categories macroautophagy, and one subtype of macroautophagy is mitophagy that targets mitochondria. And mitophagy essentially entails the formation of the autophagosome, the double membrane vesicle that essentially encapsulates cargo during autophagy, specifically around the mitochondrion. And then this mitochondrion is delivered to the lysosome for degradation. This process was initially described in yeast, but it was discovered then that it plays a very important role in human physiology. For example, red blood cells do not contain mitochondria, in addition to not containing a nucleus, being anucleated cells. They also don't contain mitochondria, and the mitochondria are eliminated through mitophagy during maturation of erythrocytes to red blood cells. This is happening through specific interactions of mitochondrial proteins called NIX. This is the first protein I mentioned to you earlier. NIX is a mitophagy receptor, as it's called. It's a protein that directs the formation of the autophagosome around the mitochondrion by interacting with specific proteins on the autophagosome, in this case LC3. And that's how the mitochondrion becomes engulfed and then degraded in the lysosome. And this is happening in erythrocytes. We don't need mitochondria in erythrocytes. They are short-lived cells in red blood cells. And also, we should not have there organelles that consume oxygen, which is the cargo that needs to be transported by red blood cells. Now, this is a physiological paradigm of mitophagy. Mitophagy can also occur in a pathological context whenever cells sense that there are damaged mitochondria accumulating. In this case, they need to eliminate these mitochondria, and this is happening through the so-called pink parking pathway, which becomes activated in the vicinity of damaged mitochondria. Pink is a serine threonine kinase that phosphorylates and activates parking. Parking is an E3 ubiquitin ligase that then ubiquitinates specific proteins on the outer membrane of mitochondria. And this is the flag that directs the formation of the autophagosome around the mitochondrion for degradation. So these are 2 paradigms of mitophagy. We found that in C. elegans, the 3 proteins that I described to you, which were isolated through the genetic screening, actually form a pathway. So pink activates parking by phosphorylating it, and then parking ubiquitinates DCT1, which is the homologue of NIX in C. elegans. in a specific lysine residue, and this signals the initiation of mitophagy. Now, having identified the genes that encode proteins playing a role in mitophagy, we can now go and specifically manipulate mitophagy away from other types of autophagy and ask what the effect is on aging or on neurodegeneration. So that's exactly what we did. We knocked down mitophagy mediators and asked whether or not lifespan of animals is altered. In wild-type animals, we don't see an effect, and this is something that one perhaps could expect, because under normal conditions in wild-type animals, we don't have induction of mitophagy. So not having mitophagy mediators would not make a difference there, although this is a paradox, and I will tell you a little bit more about that in a minute. However, in long-lived mutants, what we see is that if we knock down mitophagy, then we shorten the lifespan of these animals. We take away part of their longevity. This suggests that mitophagy induction in long-lived mutants actually contributes to lifespan extension seen in these animals, which suggests that mitophagy is elevated in these mutants. And indeed, this is something we can measure because we developed a specific tool, a specific system to monitor mitophagy, the levels and the activity of mitophagy in cells, using a bipartite reporter, fusing GFP with DS-RED, and then localizing this fusion to mitochondria. Now, if mitochondria are engulfed and then delivered to the lysosome for degradation, in the lysosome we have acidic pH, and this quenches the fluorescence of GFP, but leaves DS-RED fluorescence unaltered. So by calculating the ratio of GFP to DS-RED, we can monitor the levels of mitophagy. We can estimate the levels of mitophagy. So whenever we have a dip, a lowering in this ratio, this indicates induction of mitophagy. This indicates that mitochondria have ended up in lysosomes where pH is acidic, and that's why we have a lower ratio of GFP to DS-RED. And this is exactly what we see happening in long-lived mutants compared to wild type. There is a pronounced reduction of the ratio GFP to DS-RED, which signifies induction of mitophagy. However, if we knock down specific mitophagy mediators, then we prevent this reduction. Essentially, we block mitophagy, and that's how we can monitor mitophagy and confirm that indeed, in long-lived mutants, mitophagy is elevated, and that contributes to the longevity of these animals, of these mutants. Now, what happens to neurons when we block mitophagy? Of course, we have accumulation of mitochondria. We see this either using GFP or DS-RED or mitotracker in neurons. There is pronounced accumulation of mitochondria. Now, mitochondria that accumulate also are damaged because now there is no mechanism to eliminate damaged mitochondria in these neurons, which of course has dire consequences for these cells. So we see that there is an increase in the production of reactive oxygen species. There's leakage of calcium out of damaged mitochondria. There is also a collapse of the inner mitochondrial membrane potential that is used to produce ATP, and that's why ATP production becomes lower in these cells that cannot perform mitophagy. So essentially, we have a phenomenon of mitochondrial aging if we interfere with mitophagy. We see that mitochondria accumulate that don't produce a lot of ATP, instead produce a lot of reactive oxygen species. So these animals with inhibited mitophagy actually experience a chronic oxidative stress because of the increased ROS production, which should have, in principle, should have precipitated detrimental effects. And one of those detrimental effects would be the shortening of lifespan of wild-type animals if we interfere with mitophagy. However, as I described to you, we don't see this shortening of lifespan. Actually, there is no difference in wild-type animals if we knock down mitophagy. So this is a paradox, as I said earlier, and we would like to understand why this is happening, why we don't see a shortening of lifespan in wild-type animals, despite the detrimental effects of inhibiting mitophagy. So something must be compensating. That's the working hypothesis, the downregulation of mitophagy. So we decided to investigate further, and it turns out that the process called retrograde signaling kicks in upon downregulation of mitophagy to compensate for this downregulation. So what is retrograde signaling? It's a signaling pathway from mitochondria to the cell nucleus, where most mitochondrial proteins are encoded when cells sense something is wrong with their mitochondria. So this is a transcriptional response orchestrated by PGC1 alpha, a master transcription regulator that works together with a number of other cofactors, co-transcription regulators of the NRF family to regulate the expression of genes, to induce the expression of genes encoding mitochondrial proteins so that new mitochondria can be built in cells. Fresh functioning mitochondria can be built in cells. Now, in C. elegans, SKIN1 is a homologue of the NRF family of transcription factors that orchestrates this retrograde signaling response. So what we found is that indeed, this transcription factor becomes activated. So we have activation of retrograde signaling upon mitophagy downregulation in C. elegans. And this in turn, this transcription factor that becomes activated, in turn induces the expression of mitochondrial encoding, mitochondrial protein encoding genes in C. elegans. And interestingly, among those genes that are upregulated by SKIN1 through the retrograde signaling is DCT1, which is one of the master regulators of mitophagy. So it's as if cells, when they sense that mitophagy is reduced, they activate retrograde signaling, trying to restore the levels of mitophagy by inducing the expression of a master regulator of mitophagy. So this indeed must be compensating. And to confirm this, we assayed the effects of mitophagy downregulation in the absence of SKIN1, which is the mediator of retrograde signaling. So if we don't have SKIN1 there to compensate through retrograde signaling, indeed, mitophagy downregulation is detrimental. We see a pronounced shortening of lifespan in otherwise wild-type animals if we don't have SKIN1 there. Now, to go back to the original question of what happens in neurons, and more specifically in Alzheimer's disease models, we observed that whenever neurons experience oxidative stress, they have a way of cleansing themselves of damaged mitochondria. And this process entails the transport of mitochondria to the soma, where most of the degradative machinery resides, the lysosome system, and autophagy also is mainly performed in the soma.
I'm sorry, Dr. Tavernarakis, you have to complete.
Yes, I'm almost done. So indeed, mitochondria are transferred to the soma of these neurons for degradation. This process requires calcium signaling. So this is triggered by calcium and also requires the AMPK kinase pathway. And mitophagy is stimulated to take away the damaged mitochondria that happen to accumulate in these neurons. Now, if we inhibit mitophagy, then these neurons essentially have no way of detoxifying themselves of damaged mitochondria. So damaged mitochondria accumulate. And what I wanted to show you is what happens in two models of neurodegenerative disease, in Parkinson's disease, where we have alpha-synuclein overexpression. If mitophagy is not there, then these neurons degenerate to a much larger extent. They essentially are defenseless against neurodegeneration. Same thing with Alzheimer's disease model, where we overexpress tau or beta amyloid. These two overexpressed proteins strongly inhibit mitophagy. So inhibition of mitophagy again does not allow these cells to detoxify themselves, and they are left to degenerate. And lastly, what we observe is that there are defects also in learning and memory. So neurons that cannot perform mitophagy actually cannot function properly, even under physiological conditions, to perform cognitive functions such as learning and memory. By contrast, if we elevate mitophagy, if we enhance mitophagy, then what we see is that there is a restoration of memory deficits, of memory and learning deficits in Alzheimer's disease models. And these results have been confirmed in the mouse as well. So I'm going to conclude by just showing you the mechanistic model of the process that we believe regulates mitochondrial content in neuron and safeguards the homeostasis, the energy homeostasis in neurons by regulating both mitochondrial biogenesis and mitophagy to restore balance and take down superfluous or damaged mitochondria. And if this is not happening, then of course neurons are left without a mechanism to detoxify themselves of damaged mitochondria during aging or in the context of neurodegeneration, and eventually these neurons will succumb to death. So I will finish here and thank my colleagues and friends at the University of Crete, in the Medical School of the University of Crete and the Institute of Molecular Biology and Biotechnology here in Crete, who did the work I presented to you today. Kostas Palikaras did most of the work as part of his PhD. He's now a professor at the Medical School of the University of Athens, and Irini Lionaki, a postdoc associate in the lab, who did the biochemical analysis and the proteomic analysis that I showed you. Thank you very much for your attention, and I would be happy to address any questions you may have.
Thank you, Dr. Tavernarakis, for your interesting, excellent presentation. Is there any question very fast for Dr. Tavernarakis? Any question, please?